Welcome back to the Anatomy and Physiology playlist for your pre-lab lectures in this class. Okay, so just to remind you, these are the ones that I specifically assigned in your homework. Okay, and so what you're essentially going to do is you're going to go to the YouTube search bar. Okay, just to remind you, type in my name, Kevin Tokoph, right here. Go to playlists, go to Anatomy and Physiology lectures, and then you'll go and look at the various videos that I assign. So for example, what we're going to talk about in this particular video are two very important organelles that provide defense against various things. So the first organelle that we're going to look at is something called a peroxisome. Okay, and here's the idea behind a peroxisome is there are various reactions in humans that produce a very dangerous molecule known as hydrogen peroxide. Okay, and we're actually going to look to see exactly why it's so dangerous. Okay, and it turns out that there are several reactions in biochemistry that can produce this molecule right here. Okay, so one of the things that your cell actually does is it has an organelle called the peroxisome that basically puts a lot of the enzymes that produce hydrogen peroxide in it. So let's say you have some generic reaction, you have substrate A, and it goes to a product B, and in the process, let's say it uses molecular oxygen as a substrate, which it often does, and then out of it you get this hydrogen peroxide, okay? And we'll call this will be enzyme 1. Okay, this enzyme and others like it that do a similar reaction in which you produce hydrogen peroxide, these enzymes are localized to the peroxisome. And hopefully that makes sense, and we'll talk about why that is. Okay, so here's the thing that you need to keep in mind. Hydrogen peroxide is catabolized, so if we have hydrogen peroxide, it can react in multiple ways. Okay, number one, if we have two hydrogen peroxide molecules, okay, there's an enzyme located in the peroxisomes called catalase. So this enzyme is called catalase, and it takes these two hydrogen peroxides and it's going to generate molecular oxygen and two molecules of water. And these guys right here, these are virtually harmless, okay, whereas the hydrogen peroxide is dangerous. So these molecules are very dangerous. You don't want those guys floating around. Okay, and we'll see why that is in just a minute. But I want to think about the intuition behind having catalase inside the peroxisome. So this is a peroxisomal enzyme. Okay, it would make sense if you localize these enzymes that produce hydrogen peroxide, if you localize those to the peroxisome, it would make sense to have catalase in there because catalase gets rid of this dangerous product. So basically what happens is you have this enzyme in here inside the peroxisome, it produces hydrogen peroxide, and then you have the enzyme catalase, which is also inside the peroxisome, that converts it into oxygen and two waters. Okay, and that's, of course, two hydrogen peroxides. Okay, so hopefully that makes a little bit of sense. If you're making hydrogen peroxide in the peroxisome, it would make sense that you'd have a way to catabolize it, and catalase is the way that you do that. Okay, but the question remains is why is hydrogen peroxide so bad for you? And the reason lies in the fact of two non-enzymatic reactions that hydrogen peroxide can do. Okay, so let's actually look at the molecule hydrogen peroxide. Okay, what can end up happening is if you put this hydrogen peroxide in the presence of UV light, and by the way, UV light is something that comes from the sun. Okay, what can end up happening essentially is, see this bond right here? This bond right there, that bond between the OOs. Okay, so this OO bond is very weak. Okay, it's prone to undergoing something called a homolytic bond cleavage. Okay, and so what can happen is, recall that these single bonds are just two electrons. Right, there's essentially one electron right there. There's another electron right here, and so what can end up happening is the bond can split, and so one of the electrons can end up on this oxygen. The other electron can end up on the other oxygen, and so what you end up generating is two hydroxyl radicals. So I'll draw the 2 lone pairs on the oxygen, and then there's this lone electron right here. So this molecule right here, this is called a hydroxyl. This is a hydroxyl radical. These molecules are incredibly dangerous to the cell. Okay, they can react with DNA, they can react with proteins and membrane lipids and all sorts of things that you don't want to happen. Okay, so you'd like to theoretically get rid of hydrogen peroxide before it can, in the presence of UV light, undergo this homolytic bond cleavage. Okay, there's one other reaction it can do, so let me redraw hydrogen peroxide. And this particular non-enzymatic reaction is going to occur in the presence of iron 2+. And iron 2+ is what we call the ferrous state of iron. Okay, and you have to imagine from your general chemistry and intro to chemistry, there's some electron out here in one of the orbitals of iron, and this electron essentially is susceptible to being transferred to one of these atoms of oxygen, and that will effectively split this bond, just like we saw in the case of UV light. But now instead what you have is you simply just have one hydroxyl radical, so one hydroxyl radical with a lone electron, and then you also get a molecule of hydroxide, which has an extra electron when compared to the hydroxyl radical. Okay, both of these reactions are incredibly dangerous, and by the way, this reaction where we use the iron, this is called a Fenton reaction. This is called a Fenton reaction. Whenever you transfer the electron from the iron 2+ into hydrogen peroxide to make hydroxyl radical and hydroxide, that's called a Fenton reaction. Okay, and your other product that you get out of this is because iron transferred the electron and gave it up, it's now in the 3+ oxidation state. Okay, so this stuff right here, this is stuff I require you to know for the exam. Okay, and if you want more detail on Catalase, stuff that I don't require you to know, but if you're curious, you can go to this link here. I have a video on Catalase in Kev's biochemistry videos on my YouTube channel. Okay, so basically I've already mentioned a lot of this, but there's lots of enzymes, like I mentioned, that produce hydrogen peroxide. So that is this molecule right here, hydrogen peroxide, and these enzymes are localized to the peroxisome, at least a lot of them are, and so that makes sense because inside the peroxisome you have catalase. Catalase is inside there, and so when the hydrogen peroxide is produced, catalase can take care of it and turn it into virtually harmless products, molecular oxygen and water. Okay, and just so you know, it takes 2 hydrogen peroxides to generate 1 oxygen and 2 waters. Okay. And one of the interesting things I think that I'm not going to require you to know for the exam is that catalase can catalyze over 40 million reactions per second. In fact, catalase is known to be the fastest enzyme in Homo sapiens. Okay? So hopefully that gave you a little bit of intuition on the purpose of the peroxisome. And also notice that it has a plasma membrane as well, just like the cell does. It's one of your pretty typical organelles. Okay? So now what we're going to do is we're going to go to a different topic called the lysosome. Okay, so lysosomes also provide defense like peroxisomes, but peroxisomes provide protection against hydrogen peroxide. So peroxisomes, thus the name, provide protection against hydrogen peroxide. Okay, lysosomes do something a little bit different. Their main role of protection is against bacteria. So lysosomes, they like to protect against bacteria. Okay, that's one of the things they can protect against. Okay, so what we have here basically is something that you do need to know for the exam. I'm very likely to ask you about this on the exam. It's certainly one of the essay questions I ask you to know, okay, on Blackboard. And this is the process basically of how a microbe, like a bacteria, so a microbe, that's an example of, that's another term for a bacteria. This is the process of how the bacteria gets engulfed by something like an immune cell. So this right here, this could be some kind of immune cell, like some kind of white blood cell. You'll talk about those in A&P 2. And what ends up happening is you get a process called phagocytosis. Okay, so the actual process of the immune cell engulfing the bacteria, so that's this guy right here, The actual process of this guy getting engulfed by the phagocyte, this is phagocytosis. This is phagocytosis. So the immune cell is literally eating the microbe, okay? And in the process, what happens is the microbe gets enclosed inside this phagocytic vesicle. Okay, it's just a pretty typical vesicle that gets butted off from the membrane and comes inside the cell. Okay, and so the microbe, notice, is inside this vesicle. What's going to happen from there is there's a lysosome right here. So this thing right here, this is the lysosome. The lysosome is going to fuse with this phagocytic vesicle, and it's going to form something called a phagolysosome. It's going to form something called a phagolysosome. Now, in order to understand what happens after this, you're going to need to know a little bit more detail, but suffice it to say for now, the lysosome contains lots of acid, meaning it has a low pH. So in there's low pH inside the lysosome. There's also reactive oxidative species like hydroxyl radicals that we talked about in the last organelle. And then there's also enzymes. There are enzymes that degrade whatever was inside the phagocytic vesicle. Like certain types of enzymes would be proteases. They degrade proteins. You'll also have nucleases. They degrade nucleic acids, like DNA and so forth. Okay? And so what ends up happening is all this stuff, the enzymes degrade the molecules, reactive oxidative species damage them, low pH denatures proteins, and so ultimately what you get is basically a bunch of garbage inside this phagolysosome. Okay, and then what's going to happen is finally the enzymes are going to take care of everything. The reactive oxidative species will have done their part. The low pH will have, the acid will have denatured everything. Everything's destroyed, and so you end up with a residual body that has the leftovers. So this basically has what we call the leftovers of phagocytosis, and those ultimately end up getting discharged. So the final step is basically discharging those into the interstitial fluid, okay, and they get recycled. So these guys get recycled. Those are basically the waste products of what originally was a bacteria. So we can basically take the bacteria using an immune cell, we can phagocytize it, break it down using a lysosome, and then we can recycle the parts of it, okay? And let's talk about the various parts right now. Okay, so basically what's happening, just to recap real quick, is the immune cell is engulfing the microbe, and in the process the microbe gets enclosed in a phagocytic vesicle. Okay, then a lysosome combines with that vesicle, and the lysosome essentially dumps all of its contents into there. Okay, it dumps reactive oxidative species, it dumps enzymes like proteases and nucleases, and then on top of that, it also dumps acid into there. The acid denatures the proteins, the proteases degrade the proteins, the nucleases degrade the nucleic acids like DNA and RNA, and then the reactive oxidative species damage things further. You eventually break all that down to where it's inside a residual body, and then that stuff gets discharged into the interstitial fluid and blood. And that stuff will eventually get recycled. Okay, so hopefully you understand now the process of phagocytosis. But now what I want to do is I want to go over the biosynthesis of reactive oxidative species. And up till now I've been abbreviating these as ROS. So when you see ROS, that simply means reactive oxidative species. Okay, and the reason they're called reactive oxidative is because they're extraordinarily reactive and they oxidize things. Okay, oxygen, the atom of oxygen, so oxygen, likes to oxidize things. It likes to pick up electrons and act as the oxidizing agent. And when it picks up electrons, it gets reduced, but in the process it oxidizes things and damages them. Okay, so lysosomes, what they do is they generate a lot of reactive oxidative species. Okay, and the generation of reactive oxidative species is important for destroying whatever this guy is right here, this microbe, whatever it may be. Okay, they're important for damaging it and destroying it. Okay, the oxygen, this molecular oxygen that you have right here, this is of course inhaled from the air. Okay, so whenever you breathe in oxygen, this is the same oxygen that's used to biosynthesize these reactive oxidative species. Okay, now the committed step in reactive oxidative species synthesis is catalyzed by this enzyme right here, A, which is called NADPH phagosome oxidase. You may just refer to it as NADPH oxidase. Okay, that's another name for it. And basically what it does is one at a time it transfers one electron into molecular oxygen, and you end up with this molecule called superoxide. Notice here how there's sort of an empty space right here where you could have an electron, and that electron essentially is going to end up right there, and you see it right here in superoxide. So superoxide is one electron reduced versus molecular oxygen. Superoxide by itself is a free radical. Notice that there is I'll box it, there is a lone electron right there. So it's going to be reactive, but it's nowhere near as reactive as hydroxyl radicals. That's going to come in just a minute. Okay? So what we can do further to superoxide is we can react it with an enzyme which is denoted B, I'll box that, called superoxide dismutase. And what superoxide dismutase does is it takes superoxide and converts it into hydrogen peroxide. Now, this is a really important distinction. What is the difference between a peroxisome and a lysosome? What would be the main difference in this context? Well, the difference lies in the fact that in the case of the lysosome, they want to generate hydrogen peroxide because the hydrogen peroxide is made for the purpose of damaging the bacteria, okay, or whatever else it's degrading. In the peroxisome, they're protecting the whole cell, our cell, from hydrogen peroxide, and so that's why they express catalase. And so what can then end up happening to hydrogen peroxide, like I showed you a few slides ago when we looked at hydrogen peroxide, is I showed you that it could react with UV light and ferrous iron. Okay, that's what's going to happen here. So let me find the slide on that. Here we go. Hydrogen peroxide can react with free iron in the 2+ oxidation state, so that's this. Reacts with free iron. Iron is going to transfer one electron onto the hydrogen peroxide, oxygen, and that's going to split this bond right here, and so you end up with a hydroxyl radical and a hydroxide. And the hydroxide can actually donate an electron to something, or it can pick up a proton from solution. Okay, but either way, they're both dangerous. Okay, or you can have ultraviolet light, UV light in the case of C, and UV light is going to split the bond, and so you'll end up with two hydroxyl radicals. And the hydroxyl radicals, that's sort of the ultimate goal of all of this, because they have the potential, they're very capable of damaging DNA, they're very capable of damaging proteins, membrane lipids, and all that stuff. And so basically what the lysosome does is it uses that to its advantage to kill the bacteria, protect us from the bacteria, and then break the bacteria down into its constituent nucleotides, amino acids, and various other things that can be recycled. Some other things that lysosomes contain, like I mentioned earlier, were acid. So there are protons here. Protons, if you look at a proton, you can either draw it like this as H+, or another way you'll see it drawn as H3O+, the hydrated form. Okay. Either one of these are what we call acid, okay? And lysosomes are filled with acid, meaning that they have a low pH, okay? And the low pH denatures proteins. So the low pH, we'll say it denatures proteins, okay? And that's very advantageous for the lysosome, because what the lysosome is also going to do is it's going to release proteases. And the proteases are enzymes that hydrolyze and break down proteins. So if they're already denatured, it's going to make it much easier for the protease to hydrolyze the protein into amino acids. Okay, so it breaks down the protein key into amino acids. Okay, likewise, you can somewhat denature the DNA, and then a DNase, which is an enzyme that hydrolyzes the DNA, will break it basically down into what we call deoxyribonucleic deoxynucleotides. And those will end up being recycled. Okay. Also, there are some enzymes that are present in lysosomes that can induce apoptosis in the bacteria cell. Okay, so the things that induce apoptosis are things that they activate enzymes called caspases. Caspases are enzymes that induce apoptosis, and so what the lysosome does is it releases enzymes that activate caspases, and so they trigger apoptosis, which is called programmed cell death. It literally, another way of saying this is it's cell suicide, and specifically it's the bacterial cell suicide. So literally what the lysosomes do is they release some enzymes that activate caspases, which activate bacterial cell suicide, and that kills the bacteria. So the bacteria dies. Okay? So what we can say now, and this is something I'm very likely to ask you on the test, I particularly like learning about lysosomes, so it's very possible, so keep that in mind. Lysosomes kill bacteria in 5 ways: acidity, proteases, nucleases, apoptosis, and reactive oxidative species. This right here, if you understand how each of these things is done, you'll be golden for the test. Okay? Like I said, this is very likely to be on the test. Hopefully it makes a little bit of sense, and the nice thing about YouTube is you can rewind stuff. Okay? So in the next video, we'll talk about the mitochondria, but hopefully this video gave you a little bit of intuition on peroxisomes and lysosomes. See you in the next video.